attack that takes us beyond KIT and PDGFRA, okay? Well, what else is remarkable? How common are these baby GIST, KIT mutant GISTs in the general population? At least 30% of the entire population have them. KIT mutant little GISTs. Now, of course, we know GISTs as a clinical problem are incredibly rare, so the axiom there is that the likelihood of progression from this early KIT mutant real GIST starting point to something that comes to attention and ultimately followed by the sarcoma program here is close to zero. Why is that? This picture, it won't mean much if you're not a pathologist, but you see the little black specks in there. That's calcification, which doesn't belong in an ordinary GIST. That's a sign that the GISTs are breaking down. And the remarkable thing that we've seen in these little baby GISTs is they get to a certain size and they start to fall apart. Here's another picture. It looks all pink, doesn't it? And that's because because healthy cells stain blue, and when it's all pink, it's basically dead stuff. So they kind of get to a size, and then they start to fall apart. Another remarkable thing is that there are certain forms of KIT mutation. Some of you have heard about these. We're struggling with whether they should be incorporated into some of the risk classification schemes for GIST, because if you have a clinically diagnosed life-threatening GIST with one of these mutations, the prognosis is worse than if it's another type of KIT mutation. But lo and behold, we find these same worrisome bad actor KIT mutations in the baby GISTs that we know essentially statistically have close to zero likelihood of progression to a clinical problem. So it's an odd thing, isn't it? If it is that mutation in a big GIST that causes symptoms, it means one thing, but it's not enough on its own. In the little GIST, it doesn't tip the scale towards progression and a bad outcome. I'm just going to flip through these, okay? We also know of a few things beyond KIT that occur at the genome level, losses of certain chromosomes that occur very predictably after the KIT or PDGFRA mutations, like there's a chromosome 14, which most GISTs lose one copy of as they're becoming worse. And there's quite a bit of that that already happens at the point of these early GISTs that aren't destined to go anywhere. So we can now classify and say, well, these events, not just the KIT mutation, but these additional genetic events, they are essential to becoming a GIST, but they're not enough. They're not enough. They can represent an Achilles heel as a therapeutic target in the GIST, even though they're not sufficient in and of themselves to cause the clinical problem. Okay? And here's just reflecting back to the PI3 kinase inhibitors that Dr. Butrinsky discussed. It's been interesting in considering these little GISTs. See the brown stuff down at the bottom? This is a pathology stain that tells us the PI3 kinase pathway below KIT is strongly activated. It's receiving activation energy from KIT, telling the cells to grow and to survive. And then here's some more of the baby GIST, this little small GIST, where there aren't many cells, it's not staining brown. Again, this kind of purplish stuff is the calcification. All of this is breaking down. When the baby GIST doesn't have activation of the PI3 pathway, it does poorly. When it has activation, the cells are healthy. And without explaining it all in detail, that gives us even more of a sense that at the early stages of GIST development even, this pathway that is now being targeted therapeutically in the early stages of targeting is meaningful. Okay? So just to sum that up, we know that these baby microGISTs are on a spectrum of continuum, a biological continuum with the ultimate clinical problem. They're a very meaningful reference point that we have in very few other sarcoma types, relatively few cancer types generally. They're remarkably common in the general population. They virtually never progress to malignancy, even though they have the building block in it. The likelihood that they'll jump through all the right hoops remains low. They often involute and autodestruct, even though they have the KIT mutations. This tells us that the mutations alone, of course, are not sufficient to sustain malignant progression, and at the same time, because this means you need other gene mutations, it pushes us harder to identify those as potential therapeutic targets. Okay? Now, I'm going to flip from the baby GIST now to the clinical problem and the challenge of drug resistance heterogeneity that has already been raised. And I'm just going to draw upon one of Dr. Rout's patients who we studied, a very typical scenario, KIT exon 11 primary mutation, inoperable. The patient had a beautiful response to Gleevec with about 2 years of response, but then progressed in 4 sites, 2 of them in the liver and 2 of them in the omentum, the fat pad that surrounds the intestines. They all have the exon 11 mutation, the same starting point. And that's not a surprise because that's the first mutation that got the cells going. The problem from a standpoint of the next steps in drug therapy is that the resistance mechanism that defeated Gleevec isn't just one thing. In this patient, as is often the case, it's a number of things. When we used molecular approaches to evaluate these 4 lesions, this lettering V654A stands for a resistance mutation in KIT. It's on exactly the same gene as the starting exon 11 mutation. And this changes a part of the protein called the ATP binding pocket, where imatinib and sunitinib and sorafenib actually interact with the KIT protein. But see, these 3 down here have something else. These D820s and NA22s encode a different part of the KIT protein called the activation loop. It's the motor part of the protein. And what we can see spelled out here is that if you have this one in the ATP binding pocket, you tend to be sensitive to sunitinib, potently inhibited by the drug. But if you have any of these 3, then you're resistant to sunitinib. And this sets up the realistic expectation that when the patient began sunitinib therapy, second-line therapy, this one got smaller, but these 3 kept growing. And now we can quite well predict that. This is also a message that if a patient has a biopsy at the time of progression and you find 1 mutation, that doesn't speak for the resistance mechanisms in the rest of their GIST if there are other sites that are progressing. You know, it can be misleading to know what the resistance mechanism is if it comes from just 1 site. Now, the point I wanted to bring up is, when do these things start? I'm not going to show you the data because we're too short for time, but we are increasingly convinced, and we're really closing in on this with very sensitive approaches, that these resistance mutations that are added onto the same gene as exon 11, they don't come along when the primary GIST metastasizes, when it spreads to different locations. At that point of spread, which often involves only a few cells, the secondary resistance mutation is not there yet. But once the metastasis begins to grow, that's when just a few of the cells will develop these second mutations. We think they occur before drug treatment has started. It's not like Gleevec causes these mutations. Rather, they're already there when treatment is started in most metastases that eventually progress, and they become a survival factor for the GISTs because they're resistant to drug. It's a Darwinian process of selecting for those clones. And we're quite close to technologies where we think we'd be able to detect even very low frequencies of these resistant mutations before a patient begins treatment in their metastatic disease. Okay? I'm going to skip over this one, but just to mention that we've, in our quest to substantially expand the spectrum of resistance mutations that we've identified with Chan's help, well, I know, you're eating lunch, but what the heck, it's important, okay? I'm going to blame this on Chan, okay? This is one of his patients, and all of these nodules are metastatic GIST. Again, a patient who had responded nicely to Gleevec, but ultimately progressed in multiple locations. And what we've done, there are 46 things here. We have analyzed 46 different progressing places. This patient was progressing in many different areas. What we learned from that, we found some of the resistance mutations that we already knew, but we found, lo and behold, I think it was 6, 7 in this individual, 7 novel mutations. The value of that is that once we've identified them, we can use them as tools in the lab to ask, as new drugs come along that Dr. Dimitri's group identifies or our group identify, do they shut down the whole spectrum of resistance mutations, not just 1 or 2, but all the resistance mutations that might occur in a single individual. To show you one example of a novel one that we found in this individual, and we've since seen it a few more times. N is a amino acid, 680 is its location, and the N is changed to a K that caused the resistance. And I'll show you why. Here's a picture which shows where sunitinib in this patient would bind. There's a little hole here that the drug is filling, and that's called the ATP binding pocket. It's where ATP, the fuel for the kinase, often also binds, and sunitinib competes with the fuel to bind that. That's how it mucks up the kinase function, the KIT function. And this 680K, the mutation, basically creates a pillar, like a column, that blocks that ATP binding pocket, so sunitinib, imatinib, the other drugs that are trying to get in there physically, they have no access. ATP can still slip by, the fuel, but these drugs can't get in there. It's not just a physical thing, it changes the charge of the surrounding topography, so the drugs have a very difficult time accessing. So I'm going to rely upon George to tell us, what do you do if you have an ATP binding pocket, the usual target for imatinib, sunitinib, sorafenib, regorafenib, nilotinib, that's pretty well blocked by a mutation? Are there other ways to go after KIT, maybe other regions in KIT? Okay, So the conclusions here, the imatinib, sunitinib drug resistance mechanisms are heterogeneous. The remarkable thing is that with the help of Chan Rout, Monica Bertinoli, and other surgeons, we can catalog them and come up with a very realistic attack plan for individual patients that I think that gives us a roadmap for everybody else. So that puts us on a very, very, very proper pathway to identify the kind of drugs that are needed to shut down, again, not just a few mutations, but a catalog of mutations that might be found in a single individual. And when we talk about a cure, you know, curing cancer is not an easy thing to do, but this is the kind of information that puts you securely on that pathway. And then again, we believe that, you know, there will always be exceptions to the rule, but these drug resistance mutations develop before treatment in the GIST metastases rather than being caused by the drugs. We think in most individuals they're already there, therefore you have to deal with them rather than just say they might develop down the road and we can contend with them then. Okay? I'd like to acknowledge the clinical team that drives all of these studies. You've seen some of the members of my laboratory group who, of course, do all of the work. There are a few more of them pictured here. The GIST Cancer Research Fund has not only driven along quite a bit of work on adult GIST, but very importantly, Katie Janeway, speaking to wild-type pediatric GIST, is here for a reason because the GIST Cancer Research Fund has been very seminal in driving those pathways along as well. And I'd like to fully buttress Tanya's plea to the group. This is a remarkable organization. The GIST research has been remarkably productive. I think it's not stretching to say that the groups that the GIST Cancer Research Fund support, beyond the fact that we live, breathe, eat, sleep GIST biology and the quest for a cure, that I think, George, I mean, you'll correct me if I'm wrong, but probably every treatment that's gone through from concept to clinical trial to some sort of success has come out of the small number of groups that the GIST Cancer Research Fund supports. So there's an effort here not just to do everything, but to help those leverage the opportunity for those who are very committed to this work and would like to work a whole lot harder with the presence of funds. So please do what you can. It's the walk in October. Is it October, Tanya? Is October 9th in New York. When it's a nice day, it's lovely. When it's a rainy day, I guess it's still lovely, right? But it's a beautiful walk around the lake, a chance for all of us to catch up. And I hope that, you know, we'll collectively do what we can to support this remarkable effort that lets the researchers work harder, and whether it's joining in the walk or helping fundraise, to do what you can. Thank you. Our next speaker is one of my lab members, Adrian Marino Enrique, and I'm going to prevail upon you, Adrian, to keep it short. You know, to be a better citizen than I was, okay, and go for the heart of it. Adrian is going to, you know, he's stepping away from the broad strokes which I've addressed to give you a sense for just one of the representative processes. These days, you know, the part of the reason the funding is so important is that you can move through various possibilities in these GIST cells with remarkable facility. It can go very quickly. And the more perspectives you have, the protein level, the DNA level, the RNA level, screening for functional entities in the cells, the more opportunity you have to zero in on the bullseye, to go for the jugular if the goal is better treatment and cure. And Adrian's going to give you a sense for one of those processes.
Okay. Thank you very much, Jonathan and the GIST Cancer Research Fund, for the opportunity. So the good news is that most of the concepts I'm going to discuss have been already introduced, so this is more almost a summary of the whole day. The bad news is that in 5 minutes it's almost impossible to give even an overview of what we are doing, but I'm going to try, and I'm going to just do like a general overview and an illustration with 2 or 3 concepts that we think are useful. So we are going here today during this session from the closest to the clinical reality. We started with surgery, then drugs that are currently in the clinics, clinical trials. So now we are going a bit back, how we generate all the data that then keep us enthusiastic and that give us opportunities, how we generate the evidence to then move to development of new drugs. So as a general overview, and you've already seen this during the day, there's some precursor lesions, these microgysts, these baby gysts that develop for some reason. We are studying why, and we know a lot about that, but they grow, they first manifest clinically as a localized disease, then metastasize, and then unfortunately they become resistant to some of the treatments. So these are the challenges that we are facing and that we try to overcome. So as you know, in the case of yeast, these precursor lesions are the microyeast.